lectures, and we've had over 3,300 people that have actually physically been in the room. In addition, we record these and send them all over, well, all over the world, kind of like Jimmy Buffett and his Margaritaville radio, but we do that, and so we hope that you'll enjoy this. We have two great speakers today, and at this point, I'll let them be introduced.
Thank you, Dr. Davis. Good afternoon, everybody. My name is Russ Reed, and I'm with the National Center for the Biotechnology Workforce, and I'm a part of Economic and Workforce and Development. It's my pleasure to introduce our speakers, and our first speaker is Jason Gagliano, and Jason has undergraduate training in biology and chemistry and a master's in science education. He currently is a PhD candidate at Wake Forest University. Jason has taught middle school, high school science, physics, chemistry, and has performed research at Wake Forest University for the past 10 years in the physics department. He's published several articles in the European Biophysics Journal, being one, Journal of Chromatography, Analytical and Bioanalytical Chemistry, ABC, with over 60 citations. One research article was listed in the top 10 most downloaded articles published in 2015. For the past 2 years, Jason has been the lab coordinator at our lab at Innovation Quarter. That lab we call the Analytical and Molecular Training Lab, AMTL. And it's now part of the National Center for the Biotechnology Workforce, so we're really pleased and we welcome Jason. In addition to Jason, we have Malaysia Christian, who is a lab technician working with Jason. She aims to provide company-specific skill development and classroom faculty training. She believes in providing resources for young scientists in the broader scientific community, and which is key in advancement of scientific research. Malaysia has contributed to research in sequencing of ACE gene in relation to hypertension and antifungal drug development. She holds a Bachelor of Science in Biology from the University of Arkansas, Pine Bluff. Location, she has worked with a variety of companies, including research quality assurance and environmental management, and some of these companies include, but are not limited to, PSC Environmental, Pace Analytical, and most recently Pepsi Bottling Ventures. She hopes to continue to provide scientific research opportunities and training to young scientists, sparking the interest of their needs and minds. So with that, I want to welcome our speakers, and I want to welcome you all to this SciTech, as Dr. Davis has said. Speakers.
Make sure I'm not talking too loud. Can everybody hear me in the back? Okay. Well, thank you, Russ and Dr. Davis. Thanks for letting me give this talk. I'm the lab coordinator of the Analytical and Molecular Training Lab. It's at Forsyth Tech in the Economic Workforce and Development Small Business Center over at 525 Vine Street. And this is a snapshot of the lab. So it's a very nice space. You can see that there's tons of equipment in here, state-of-the-art equipment, and they're broken down into 4 sections. This is our molecular biology, next generation DNA sequencing section. Also can do forensics. And then we have gas chromatography, liquid chromatography, and then we have mass spectrometry. And then the left side of the lab, we have microscopy, and then areas where people could do sample prep. So that's just a brief snapshot of the entire lab. So the AMTL is based on 3 pillars, and that's education, training, and a lab resource. So in terms of education, we offer courses where we go into scientific theory. We also have some outreach programs. In terms of training, those courses will always have a practical application to them, so folks can get trained on very high-tech equipment. And we also can train in standard operating procedures and train people in following those procedures. And then it's also a lab resource. Excuse me. So if you were a small company and you had a product that you wanted to develop, but you didn't have hundreds of thousands of dollars to get a lab infrastructure and to buy the high-tech equipment you need to build that product, well, you could come to the AMTL and actually use the equipment there to develop your product. And so it's a very nice resource for small businesses in the science area. And you could also rent bench space, and we also offer customized method development where we try to help these ones actually develop the methods they need to develop that product. So in terms of the vast amount of equipment, we have high and ultra performance liquid chromatography. We have gas chromatography, 2 different kinds of instruments. We have Agilent and Shimazu. And then we have gas and liquid chromatography mass spectrometry, both in a single quad diode and a tandem quad diode. Now, a lot of folks may not be too interested in chromatography, so I did a little demonstration. And so basically, chromatography is literally just the science of separating a mixture so that you can analyze the individual components. And we'll see if this makes that point, this little illustration. So we have our sample, we have the stationary phase and our mobile phase. So the sample is a black marker. I'm going to put it on the filter paper here. And so one question you can ask yourself is, do you think there's more than one component in that black marker, or is it just one dye that makes it black? Well, the only way to know is to do chromatography. So what you can do is put the filter paper in the water, and then what will happen is the water will move up the filter paper. That's why it's called the mobile phase. And then what you'll see is that as the water moves up the filter paper, different colors are coming out. So you can see the blue at the top, maybe a little bit of purple or yellow in the middle. Might be hard to see. You might be able to see that blue a little bit better. But that's basically chromatography, where you take a mixture and you have a stationary phase and a mobile phase and a detector, and you separate out the individual components so that you can analyze each individual component separately, and you can get information. So this simple experiment actually gives us some information. So we see that there are more dyes, maybe about 3 dyes we could see in there. This blue dye up here, did you notice it traveled further up the filter paper? So most likely that dye is probably polar or even charged because it has a greater affinity for the water, whereas the dye that stayed down here is probably nonpolar, so it's hydrophobic and has a greater affinity for the filter paper. So not only can you separate them out, but you can get some information about the individual molecules, or in this case, dyes. So the molecular biology equipment we have real-time PCR. PCR stands for polymerase chain reaction. It's a Nobel Prize winning technique that was developed in the 80s and really helped forensics. So when there's a very small sample of DNA at a crime scene, they use PCR to amplify that DNA, even one single molecule of DNA, into billions of molecules that you can then do a genetic analysis on. So it was after PCR that we were able to actually do those type of STR analyses, DNA profiling at crime scenes. But this is a real-time PCR machine, which means that you can actually watch the amplification in real time and see the curve. And by having that curve, you can actually calculate how much of the original sample was there. And then we have a spectrophotometer that's called a nanodrop. It's a nanodrop because you can literally just take 1 microliter of sample, put it on there, and do a spectrophotometric analysis on it. So maybe some of you have done spectrophotometers in the past where you have a cuvette that's about 1 mil So that's 1,000 microliters. So this could do just 1 microliter of sample. Then we have a fluorimeter and a bioanalyzer. Now the fluorimeter and the bioanalyzer work off of fluorescence, and fluorescence is a lot more sensitive than spectrophotometry. It's about 1,000 times more sensitive. So this could measure proteins, glucose, RNA, DNA. This does DNA, RNA, and protein, gives you the size of the fragments as well as quantitates it. And then we have our next generation DNA sequencer that can sequence the human genome. So all the equipment I showed you is really state-of-the-art. The reason why we shoot for the state-of-the-art equipment is because when we're doing education and training, we want people to have the equipment or be trained on the equipment you're most likely going to see in an actual business. For example, Illumina, who does this sequencer, has about 80% of the market. So getting trained on that, you're more likely to see an Illumina instrument in some type of business after you've gotten that training. So some activity in the lab. We've done tours. Roughly 14 different companies have come through, and roughly about 100 people. We've done equipment training. You can see the different equipment broken down by hours. So about over 200 hours of equipment training. We have some bench space rental that's in negotiation with 2 companies currently. We do a lot of staff professional development. We've done about 13 different events, roughly 100 hours. The reason why we still do professional development is because the better we could become, the more of an asset we could become to our clients. So the more methods, the more insights we can get into these techniques, we'll be able to help clients develop their products and be successful. In terms of developed materials, we've got about 3 customized methods that we've made for clients, and roughly 150 standard operating procedures and 150 different logs. And then in outreach, we've had high school students, middle school students, teachers, for SciTech students, Wake Forest students, faculty and staff. So roughly a total of 100 folks that come through and about 100 hours. And this was roughly 2017. So Dr. Davis brought his forensic chemistry class in one day, and Dr. Davis and I came up with a small lab where the students got some mock drug samples, 2 mock drug samples, and you can see the vial here. And then they had a chain of custody form that they had to fill out to check the drug sample out, and they had to make sure that they brought the drug sample back and checked it in. If you're a forensic scientist, you don't want to walk away with the drug. That would be bad. And so what they did was they used gas chromatography mass spectrometry to analyze the samples to see what molecules were in there. So the first group analyzed or discovered these, so caffeine, lidocaine, and tetracaine. And then the second group discovered these in the sample, niacinamide, benzocaine, and guaifenesin. And then this is where we were just kind of discussing a little bit of the theory. The mass spec will give you fragmenting patterns that you can predict through the bond energies. And so we were looking at what the bond energies looked like in some of the individual molecules and what we would expect to see in the masses. So the students really enjoyed this lab from what I know, and you can see the practical application of being able to take a sample and tell you exactly what molecules are in there. And then we had a genetic class come in where we did DNA quantitation, also for SciTech. And they were doing DNA quantitation with 3 different instruments, the nanodrop, the qubit, and the bioanalyzer. So here's what a bioanalyzer trace would look like that they got. You got fluorescent units on the y-axis and then base pairs of DNA. And what's nice about the bioanalyzer is you got 2 peaks on either side that are internal standards. And then the peaks they identified was this 83mer? and 157. And so then what the instrument can do is integrate those peaks and give you the amount of DNA for those individual peaks. And a neat thing we did in this lab was, without the students knowing it, because we want to make it a real-world training, I secretly put in some contamination into one of the samples. And so the numbers between the nanodrop and these two instruments were way off for that sample. And so then we had to discuss what could be the problem. And we came to the conclusion there was contamination. It turns out that the spectrophotometer is very sensitive to contamination. It will measure anything that absorbs at 260, whereas the fluorescent methods are more specific for your molecule of interest. And so these would be good if you think there's some contamination in there. And it was a nice real-world application of what you might see in industry. So we've had middle school students and high school students come through the lab. So Stokes County students came in, and what we did was we told them to take their cheek cells and their spit and put it in a vial, and then they extracted out their human genome from their cheek cells. And so the kids were very excited with their test tubes to run home and show mom their human genome. And then we had the Northwest Cabarrus High School students come through, and we did DNA gel electrophoresis. So you can see, maybe you could see there's the kids huddling around the gels on a transilluminator with their smartphones out, and they're taking pictures of the fluorescent glowing DNA in their gels. So at the Innovation Quarter, where we're located, we're kind of flanked by Wake Forest. So right across the street's Wake Forest Biotech Place, and then right next to us is the School of Medicine, also Wake Forest Chemistry and some of the biology department. And so, of course, they come through and would like to use some of our resources. And so here's Dr. Ravi, who is studying the microbiome. So he's literally working with feces daily and looking at the metabolites that the microbiome of individuals are producing, and then using GC mass spec to identify what those metabolites are. And then Dr. Bonin of physics is doing a drug discovery project using the next-gen sequencer. And then we have a graduate student who's looking at iron-sulfur cluster proteins and their association with ribosomes. And so we were helping her develop a method to be able to get that off the ground. So the lab is not only a part of SciTech, but also a part of the National Center for Biotechnology Workforce. This is a snapshot of that homepage, and here's the URL. And so the National Center has this amazing yearly program called the Biosciences Industry Fellowship Program, or BIFP. And what it is folks from all over the nation can come in and travel around North Carolina and see the biotech industries in North Carolina and what they're doing. And so here's the BIFP folks from 2017. So they came into our lab, and what we did was we did next generation DNA sequencing with them. Incidentally, in your packets, there's information about the BIFP, because if any of you were interested, you can go to the web page and put in an application if you wanted to do it. This year it runs from, it's a 3-week course, so June 11th through the 29th, but some of that information is in your packets. So like I said, they came into the lab and we did next generation DNA sequencing, and I just wanted to show you some data from that sequencer. So the sequencing is done on what's called a flow cell, and it has 4 lanes. And DNA is double-stranded, and so what the sequencer can do is it can sequence in one direction, and then it can sequence in the other direction. And so for each lane, you have read 1, which is one direction, and then read 2, which is in the other direction. And just to show you how much data comes out, this is 94 million reads, 94 million reads. They're about 150 characters or 150 base pairs. 93 million, 93 million, 94 million, 95 million, for a total of 108 billion base pairs of data. So to put that in perspective, imagine if you wrote on a piece of paper a word that was 150 characters, or a sentence with no spaces, 150 characters, and then you copy that 108 billion times. That's how much data comes off this machine, and then you got to make sense of it all. You got to do some bioinformatics. So we did some bioinformatics, some introduction to bioinformatics, and so what they were sequencing were two viruses. One was a bacteriophage, Phi-X, and the other one was adenovirus. So adenovirus got its name because it infects the adenoids, and so it gives us the cold, and it also gives us pink eye. So my wife right now has adenovirus because the antimicrobial drops aren't working, so most likely it's a virus that she has. So adenovirus, this adenovirus species is called DL309 and was never sequenced before, so I thought it'd be cool for these folks to sequence it. And what we did was, these are the reads that come off the DNA sequencer, and this is the genome of a known adenovirus. So adenovirus is about 35 kilobases long, so 35,000 bases. And this is the coverage that we get from the reads in each section of the genome. And you may notice that something stands out here. As you get to this section, there's absolutely nothing. So this mutant has a huge 400 base pair deletion in this one region, and it really stands out. And so that's an interesting find about this particular virus, and we'll have to do further analysis to see, what else does it mean? How does it affect its phenotype? But this was just a little intro to bioinformatics that the BIFP folks did. We've also had high school students in the lab, so this was a week-long Biotech 101 course. And what the high school teachers learned was very simple, cost-effective biotech experiments that they could take back to their classroom and start introducing biotech to their students. And that is it for me. So now we'll invite up Malaysia Christian, our lab technician, who's very brilliant, and we look forward to hearing her. Do you want this? Or do you want to use the mic? Okay. Let me just turn this off.
Good afternoon. Can everyone hear me? Okay. Good afternoon. My name is Malaysia Christian, and I will just be telling you about some of the training and workshops we plan to offer in the upcoming semesters at SciTech's Innovation Quarter. So, our vision is to create scientific courses that can help personal and professional development, to provide the skills and the support that are necessary for the industry, to create a talent pipeline for our science partners who need individuals in the laboratory, and above all, to provide our students with the ability to have long-term employment. The AMTL is continually creating courses that will help our students and the industry. So these are just a few of the courses that have been developed and are in the process of being coded. So our core laboratory skills class is a class that was created to do basic fundamentals in the lab for entry-level technicians. Our next generation sequencing lab is for next generation sequencing intro to operation, data analysis, and interpretation. We also plan on offering a number of intro courses in bioinformatics and chromatography, and our customized courses are for our clients who have specific analysis and techniques. So the core laboratory skills course was created from the National Core Skills Standards in conjunction with the C3BC grant. So different laboratories have different techniques. which they need to train all their new employees on. But regardless of those techniques, the fundamentals are in every lab. So a set of core technical skills was created because those fundamentals are what gets students hired, because they'll train you in the more complex applications. So the core skills standards were created to answer the question, what do all bioscience technicians need to know? And what do they need to be able to do to work inside the industry? So these skill standards were created to help our students better meet industry requirements and prepare to be successful in that workplace. So, skill standards are skills, abilities, and knowledge that each person needs to know in order to go into the workplace. It's created of 5 different parts, and that's critical work functions, key activities, performance indicators, underlying technical knowledge, and assessments. So, the core laboratory skills course, it's a 10-day course. It's for individuals of various scientific backgrounds. So you can go in little to no knowledge, or you can go in as a senior technician and get a refresher course. So some courses are only classroom courses, but this is a hands-on course also, and that's what separates us from traditional classroom courses. Some techniques in the lab, such as pipetting and titrations, are better learned through hands-on. Combined with our small classroom size, we can better assess each individual and provide additional training. The 9 components are personal protective equipment, measurements, glassware, balances, pipettes, solutions, pH, titrations, and autoclave and sterilization. Most of these do have hands-on performance indicators or assessments. Our main objectives are laboratory safety, metric conversions, calibration and verification of instruments, collecting and analyzing samples and data, the importance of controls, and cleaning and sterilization procedures. So during this course, students will perform some key activities, including selecting proper personal protective equipment in various scenarios, using the metric system, preparing solutions and equipment for use in various operating procedures and recording procedures. So some of these performance indicators include properly calibrating the balance and the pH meter, setting up titrations, selecting the correct pipettes and tips if applicable, and identifying different glassware. So all of these are hands-on. All of the students will receive equipment of each of their own, and they will take a assessment that will be given by us. So why should someone take core laboratory skills? Possessing these skills make potential candidates much more suitable for laboratory positions. This allows potential employers to identify individuals who have received relevant training. And we are molding our students into high potential employees whose skills have been well monitored and assessed. If you would like to sign up for any economic workforce and development class, you'll be able to find the course catalog online at SciTech's website and the course numbers, and you'll be able to sign up in person or over the phone. The next steps for our lab, we have already submitted a grant to the system office, which we hope to be hearing back from soon. We plan to submit future grants with the North Carolina Biotechnology Center. We will continue to work with our business partners and on their projects to help those succeed, and we will build more partnerships with our local industry clients and to co-develop these courses so that we may start offering them. Thank you.
Questions?
Malaysia, thank you so much for coming and talking with us today. I thought you did a great job. I may have missed this, but if I have students who I feel could benefit from learning how to write SOPs, do you have anything that might help them?
We are continually creating courses, and that is a very good idea. We do write a lot of SOPs, so it would not be difficult to create a course from that. So I will take that suggestion. That is a very great suggestion.
This was very interesting. I noticed that you said, or rather, the first speaker mentioned Cabarrus County, excuse me. Struggled with that for a while now, which makes me think that where it's, an hour and 15 minutes from here, it's much closer to Charlotte. Are we really kind of moving ahead of Charlotte in this kind of technology?
We are one of 58 community colleges in our area with some of the equipment that we do have. So that's why a lot of teachers would rather bring their students out to our lab than to one in Charlotte. We are continually keeping up with new techniques and new instruments.
Thanks for your participation. It's been very open in the information for things that most of us might not know what is going on here for SciTech. So pretty good job. So my question has two parts. One is, what will be the prerequisites that a student might have or bring to you in order to then take the core lab skills classes? And the second part of that question will be, what will be the associated costs to the student who might be interested in also taking those classes?
Okay. To the first part, there is no prerequisite to the class since it is an entry-level class, but we do send out a short assessment to get to know what level students are at so we can better prepare for when they come in the class. So everyone, when they sign up for the class, an assessment is sent out, and then based on that assessment, we'll see where their skills are and how we can better help them, even if that means making a smaller class size to better help them. The second part, as far as cost, there is not a cost on it just at this moment. But for SciTech students do receive a different price than outside individuals. So once a price is up, for SciTech will, and all its instructors will, it'll be available to all of you. Yeah.
Okay. If there is no money, you can answer, but if there's money, maybe not.
Well, just to give you an idea. You guys hear me? Let's talk about what we just learned here today. This is awesome. You guys realize, in the audience, what this is. What this is in clinician is a lot of things. First off, Russ took $15 million federal dollars, right? And with the help of about a dozen other community colleges across the United States. Did you have one in Hawaii? No, we had a visitor from Hawaii. Oh, okay. So why? I don't know. Why did I get to go to Hawaii? To develop these skill standards. And so ideally, this is the national skill standard. This isn't for SciTech. This is national. We just started here. Right? Then you add this equipment. So what I'm hoping to see is, yes, I hope we're ahead of Charlotte. Now, granted, the dole money from Murdoch, you have to do a lot to get ahead of the dole money, right, in the Murdoch campus. But I think here we have opportunities. Russ has told me that you've talked to people that said this is one of the nicest labs in North Carolina. Right? I'm going to puncture some egos right now and hurt some people. I'm going to throw a left hook. Jason's left, and I apologize for that because his son had a little accident. He needs to go take care of that. So that a lot of our clients, it's Wake Forest. Wake Forest is coming to us because of this equipment. So here's how I'd like to see this equipment used. One, if you're a student at SciTech looking for a 2-year degree, like an associate's degree, AAS or whatever, I'd love for you to find a way, for us to find a way for you to work in this lab. Because if you know how to use this DNA sequencer, or you know how to use some of these other things, you're probably worth more. And my job here is to make sure you leave here with a career, not a job, a career. And so we're hoping that we can help you with that. Two, I'd love to see more science in our area. We have Innovation Quarter. The downside to science in North Carolina is it seems to be in one place, which is Raleigh. But Raleigh is not the only place. All right? There's Winston-Salem and Innovation Quarter, and we are growing with the help of this lab, with the help of the national certificate, and hopefully with the help of our businesses, we can recruit more companies here. We may not start off by recruiting companies that have 10,000 jobs, but there are a lot of really smart people here, in part because of Wake Forest, that can start a company and grow a company. And we could be the place that happens. If we have a workforce that can work with these highly complex machines. I'm not a scientist. Going back to the money, here's my problem. Some of those machines, I flipped the switch, I just lost $2,500. DNA sequencer, is that not correct? Right. So I've got to find a way to cover that $2,500 loss every time I turn that machine on. So I'm hoping to do that through grants, through partnerships with local industry. And sometimes maybe we have to charge for that expense. So there's going to hopefully be a different cost for students versus the outside world. A business may be able to afford $1,500 a head to learn how to use one of those machines. DNA sequencer is what I'm talking about. Our students, on the other hand, I don't know if you guys are like me, but you don't have $2,500 laying around, or $1,500 laying around. So we've got to find a way to build that into our cost, whether it comes from the school, the government, private industry helping us out. That's why we're still working on it. I'd love to get the cost down to. Ideally, I'd love to get it down to free. Whether or not that happens, we shall see. But at the very least, get it down to the what we call occupational extension cost, which means typically a class will cost you about $185. Right? That I think is doable, and we may be able to get some business partners to help us out if we go down that path. So I've got the scientist, and then I got to handle the money side of the equation, which is how do I get it paid for? This is a great opportunity for us as a town. I'd love to be the next Seattle. I drink a lot of coffee, but I'd love to see us be very innovative. If you ever go to Washington, it's an interesting place. They hate businesses that don't start there. Right? You hear that? They hate businesses that don't start there. My job as an economic developer is to recruit businesses. You guys ever heard of Amazon? Right? And so they are touting that they're going to put 50,000 jobs somewhere in North America. So we as a region have submitted our application and told them how great we all are. And I'm sure we're one of thousands of towns that have done that. Right? But what's interesting is that states like Washington did not want that. It didn't start there. Microsoft, at least in their eyes, started in Washington. Who else started in Washington? Was it Starbucks or Seattle's Best Coffee or whatever? They're big on growing their own. I think we could do some of that here too. We are the city of arts and innovation. Now, I've gotten up on my soapbox here and probably said way too much. Any questions while I'm up here? How are we doing on time? Okay. Let me ask you a question. I'm gonna put you on the spot. So you're a local employer, right? You've seen a little bit about this. What if I were to go to local employers and say, if one of our students takes this class, this 10-day class, and learns the basics of how to work in the lab, could I go to an employer and say, I want you to guarantee them an interview? Not guarantee them a job, but guarantee them an interview?
I'm Bart Clayton. I'm a technical director at Piedmont Animal Health. One thing I want to go back to was the point that this gentleman raised about technical writing skills. This is one thing that we have a company of about 25 people, mainly high-level scientists, and we do a lot of our work outsourcing and using laboratories, and some local and some in Raleigh. But we're always looking for an extension where we can, get a quick answer. So to answer your question, I think we would have to have a bigger discussion about that. What I do like, what I'm seeing is that there's, really an interest in science in this area, and I've always been very frustrated with everything being in RDU. So the more resources that even a small company like Piedmont, if we have just having this knowledge now will make me want to see this laboratory, and then that starts a discussion, and you can learn more about what our industry really wants, And yes, it is about basic lab skills in some ways, but in a drug development industry, there's there's a whole regulatory world that you have to navigate. And what I see as a kind of a deficient area is knowledge about good practices in manufacturing, and in particular GMPs. That to me is, our banners and Patheon now, they seem to have their own world. And, that's a conversation I think we could have. So I'm very impressed with the level of equipment, and I'll figure out a way to talk to you guys.
Have you down. And we are working on GMPs, if I'm not mistaken.
Yep.
It's a little bit different. So if you guys are familiar with good manufacturing practices, come on down. And the FDA is probably a whole other set of classes that we could probably be offering. But we're making our way. Oh, I'm sorry. Oh, we're just talking about, yes, we do offer some GMP classes. Probably need to do a little bit more about technical writing. So if you guys are interested in the sciences, you heard it from the horse's mouth, so to speak, technical writing is a skill they're looking for. Any other questions? Yeah, go ahead, Russ.
We've just put the National Center has put in a grant actually to develop a short GMP course. So we're hopeful to hear back in the spring whether or not we'll have that. In the meantime, we'll probably develop the course in any way, but we're hoping that the National Science Foundation will help support the course.
You might need a speaker for that course.
Yeah. Great. Thanks.
Malaysia, back to you.
Any more questions?
Hi, I'm Judy Berry with North Carolina Biotechnology Center, Piedmont Triad Regional Office. This is a wonderful asset for this region, not just Winston-Salem Forsyth, but for the Piedmont Triad region. And I noticed that when Jason was speaking, he talked about some tours that included middle school and high school students. I am forever getting calls from all over about bringing classes, and they think that I'm the headquarters and we're just a tiny two-person office. I said, there's not much to tour here, and I did not know if how are you going about that if you're letting middle school and high school come to your facility? Is that just in Forsyth County? I noticed that, you've got the Greater Charlotte Region's Community College, and they had a teacher that was involved with the course, the fellows course. And would this be a route for me to go when I get these type of calls to direct it to you if you have educators, especially your science, biology type teachers in life science, Stokes, I get them from everywhere. And could I direct that to you, or you I don't know how you're looking at that, since you're also trying to make money off of it too. For industry, because a business, by training their employees, they're going to make money, and it's a write-off for them to educate their employees to come to this. So that's just a question that would help me in directing the many calls that we get.
So Judy, the answer is you can direct them towards us, and we're going to take the instructors. We want to talk to the instructors first, obviously, and we'll take them in small groups. We've been doing that now for a few years, and it works out very successfully. So yeah, and we've had them through our fellows program. We've had referrals, and yes, you're right. And so please do, yeah. And you're right. Once we get a little bit more commercially viable, we'll be a little bit more selective about doing tours and stuff. Does that make sense?
For the entire region.
Oh yes, yeah.
It's for everyone. Like,
Yeah.
we're so glad to see Piedmont Animal Health be involved because that's jobs. That's jobs.
So Judy, you have to come up here now.
Okay.
Yeah, you're going to thank our speaker.
My pleasure. As one of the sponsors for this SciTech Lecture Series, the North Carolina Biotechnology Center Piedmont Triad Office is very appreciative to you, Malaysia, and to Jason. And we're continually learning. Our office is continually learning. We learn from people like you and Jason for SciTech, the leadership. We're so fortunate in the students at Forsyth Tech. You have no idea how fortunate you are to step foot on this campus, to go to the different classes, your biotech classes, to learn, because you're creating your foundation for your future. And it's a delight for me to see these assets get to be promoted publicly in this forum. And well, thank Russ for all these good lectures that you provide for free. Not many people do that. And we want to thank you so much for being part of this and for everyone that's attending today.
If anybody wants I have some copies of the skill standards here, so if anybody particularly wants a copy, I'll happy to give it to you.